Hello, everyone. Welcome to Chapter 1, Part 2. In this chapter, we're going to pick up with Theme 5, and we're going to be talking about evolution and scientific method. So, in this, what we're going to begin with, though, is an idea of taxonomy, classification. I want to talk about the domains of life and the kingdoms. We want to talk about unidiversity of life, and we want to talk about Darwin. We'll talk about the tree of life a little bit and get an idea of what's going on there. So that's what we're going to be looking at. Now, the idea we got to start off with, because we are going to be discussing taxonomy, is this idea that life may have evolved from a common ancestor, and that could be something difficult for people to accept, that life evolved from a common ancestor, that organisms today. Now, let's kind of think about this in such a way. Let's say that I was looking at cars, and let's just say you didn't know very much about the history of cars and you only saw the modern cars that we have today, but you didn't really see like the very, very old cars. And I'm talking like the very first cars, like the 1898 models, things like that, late 1890s, early 1900s. But before there were cars, there were horse and buggies. And the very first car were actually called horseless carriages. And what they did is they just removed the horse and they put an engine in a carriage and propelled the carriage. So they basically took a buggy and made a car out of it. And that became the foundations to build cars was horseless carriages. And a lot of cars today they are going to be kind of descendants of those very first prototypes of horseless carriages. And so if we look at it, we have four wheels on most cars. You have some of the characteristics. You still have why there is still a trunk in the back, why there are certain things, and why certain things are called what they called are because that they came from the original carriages that we base our cars on. Now, with that, guys, I want to kind of help you understand that just like how that happens, if you look at cars, trucks, SUVs and all that, they point their way towards a common ancestor because they're all built in a generally common feature way. Well, so with vertebrates, for example, seahorses, jackrabbits, and birds, they have differences, but when you look at them, their skeletons are arranged in such a common way that this very well points to a shared common ancestry because that common ancestor was like a prototype in which we descend all future generations of species from. Now, if we modify from that common ancestor, and the fossil record shows us this really well, there's huge amounts of evidence in the fossil record showing us the details of how life may have evolved and genetic evidence as well. And one of those things that really can fly in the face of faith sometimes, of course, is this idea that 3.5 billion years ago life began. And again, you're never required to, like, I am never, like, I may ask you, according to science, when did life originate? When was the origin of life? But I do not, like, I'm never going to make you guys answer questions as it says, like, well, true or false, God exists or not. And you would have to say that to be right, and then I would be like, "Oh, you're wrong. This is the right answer," and then make you go against your beliefs. But you need to understand what it is that we're teaching. So some things there. Now, taxonomy. Human beings love to classify. And if you don't believe me, think back to high school. In high school, what did you do? You'd see somebody, you might see this person, the way they're dressed, the way they act, things like that. Say that person's a nerd, or they're a jock, they're a cool kid, what have you. This person, they're emo. And you would look at the characteristics they had, how their behavior, how they presented themselves, how they act, how they looked, who they hung around with. And that's similar to what we do in taxonomy. Is it is a classifying, formalizing, naming, putting organisms together with groups based on their characteristics and their degree of evolutionary relationship. Because basically, um, there is going to be an increasing breadth, where at the top of the list, you're going to have more organisms than the bottom, but we go from more to less inclusive. So at the top, they have less to do with each other, and at the bottom, they're going to have more to do with each other, more similar. And so this also tells us if I go back far enough, I'm going to be able to find out that I'm related to all life on Earth, and I find that deeply um, um, humbling. Um, I see that as a thing that people should really kind of consider more is this our connections to we are not above the animals, we are another one in science's view, in my view. Now, what I want to help you guys understand, and you can easily remember these from the mnemonic of Do Kings Play Chess on fiberglass stools, is domain, kingdom, phylum, class, order, family, genus, and species. Now, if I look at a funnel, imagine a funnel, just think of a funnel, and at the top of the funnel, that's going to be domain. As we go just a little bit down in the funnel, it gets smaller. That's kingdom. A little bit further down is phylum. Just a little bit further down more from phylum is class. A little bit further down is order. A little bit further down is family. We're getting near the bottom now, genus. And at the very bottom, there's a hole. And out that hole comes a single species. This would be a funneling sort of thing done by taxonomy. So we could say that if I have a species, a single animal, that a genus is nothing more than a group of related species. And a family is just a group of related genuses. And an order is a group of related families. And a class is a group of related orders. And a phylum is a group of related classes. And a kingdom is a group of related phyla. And then domain contains all these kingdoms, and life contains the three domains of life. Now, herein lies the key principle to understand is that in life, as we try to taxonomically classify this, we go from most inclusive to least inclusive to where we get to a single species. And so, gentlemen who are listening to this class, if you guys are out in a club somewhere, you see an attractive lady and you want to go up and talk to her, just walk up to her and say, "Girl, your name must be phylum because you're above class." So another way to remember some of these stupid things. But as we think about this, you are using characteristics and evolutionary relationships to put them in there. Now, we'll be talking about throughout Bio 2 the different phyla, and we'll talk about some classes, and I'll even talk about some orders. When I talk about amphibians, I'll get into the orders of amphibians, like order Caudata. We'll talk about some of the different vertebrate classes and things like that. We don't really ever go into family, genus, species, but we do go as high as the order in this course. And I will mention some specific species. But let's use an example. Let's say I have a whole bunch of living isms. Now, these things are all eukaryotic. They all have eukaryotic cells. But some of these are, for example, protist. Some of these are fungi. Some of these are plants. Some of these are invertebrates, like sponges and things like that. And then some are vertebrates. Some are bears. Now, if I wanted to classify bear, I need to first go with the domain of eukarya and say everything who has eukaryotic cells. Okay, now let's eject anything that is not an animal. That goes bye-bye protist, bye-bye plant, bye-bye fungi. But we get to keep sponges. Then we say let's get the phylum of Chordata, which might include the vertebrates. We can get rid of these guys here. We can get rid of all these guys here because they are invertebrates. And then we're left with these guys who have backbones and stuff. And then I say mammals. Well, let's get rid of amphibians, reptiles, birds, and fish, and have just the mammals. Animals with fur, animals who have mammary glands and things like that. Most of them give live birth, but not all. All right. And then we say, well, of those mammals, let's go to the ones who eat meat. And then we can get rid of porpoises and zebras. And then we can say, let's get rid of all these guys who are felines and canids, and let's go to the Ursidae family, the family of bears. Now, Ursidae here, for example, spectacle bear is not, and also pandas, they are in the family with like grizzlies, black bears, and polar bears. But in the genus Ursus, and these guys can breed together. You can get inbreeding between some of these species of bear of the genus Ursus, and some of these actually do inbreed and hybridize. Is we got to get rid of pandas and spectacle bears. Then we're down to just the one species, Ursus americanus, the American black bear, like Smokey the Bear. That's an animal here in Tennessee, where I'm currently living, and that's where we are. Most of my students listening to this are, but this is also going on YouTube. So some of the people may not be from, they may not know Tennessee, but we all know Smokey the Bear is the one that's famous, correct? So now, when we think about that specific name that I gave it, I said Ursus americanus. This is what is called a scientific name. Now, all people say, well, why do you use scientific names? Is it just to sound smarter than everybody else? Is it to be pretentious? And I'm like, no. This is so that I can, guys, give a very standardized name. Let's say I'm working with someone who lives in China or India or Russia, someone in Europe. And though they may speak English, they may not know the individual animals that we are accustomed to here. Now, a great example of this, just kind of quick example. When I was in graduate school, I was studying movement patterns of a particular salamander. And this salamander that we were moving, I wanted to use real-time kinematics to study its movements. So I went and talked to some people, and one of the instructors was from China. And now she did not know what a salamander really was, though in China they do have salamanders. They have some of the largest salamanders out there in Asia. So but that was the thing, is she may not be able to know, like here, somebody might call them some of these animals like spring lizards, which they're not really lizards. Or they might call some of the different species things like snot otters, which is the hellbender, which that means nothing to anyone who lives anywhere else other than here. That's so if I said Cryptobranchus alleghaniensis, well, that's to separate it from any other cryptobranchid species out there. Now, those names are standard, and it helped me to communicate with a scientist who was not from here. Now, it is part of two names. That's why we call it a binomial nomenclature. There's two names, genus name and a species name. Now, the genus, you always capitalize the first letter of the word, and the rest of it is of course non-capitalized, and you always write it in italics or you underline it. In this case, I'm choosing the spotted salamander, which is its common name, and the genus is Ambystoma. Now, Ambystoma maculatum is only one, but there's Ambystoma opacum, Ambystoma talpoideum, Ambystoma mexicanum, Ambystoma opacum, talpoideum, tigrinum, mexicanum, etc., 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 etc. There's just a handful out there. And what you'll see, guys, is that the second name, the species name, sometimes called the specific epithet, this is all lowercase, always lowercase, and you either italicize or underline it when you write it. Now, if you're handwriting it, you'll underline it. If you're typing it, you'll take it and italicize it. And I use Ambystoma maculatum as my example. Now, but that is how we do it. Now, one of the things I may require you to do is recognize an appropriate or properly written scientific name. It is something that we use and we do in science. Now, memorizing them is something people do. Loads of scientists who are really into a particular group, like amphibians or something like that, or reptiles, they memorize those scientific names and they discuss them with people. They seriously get into it. And I will say herpetology is a great hobby. I will let the class know when I have my next naturalist rally coming up this fall. And if you want to go out and see some salamanders with us, those who are in my honors class or in our class of mine who would like to go out, let me know. And I'll let you guys know when it happens. It'll be happening in the fall. Hopefully, COVID doesn't ruin it because things have been ruined by COVID. All right. Now, what we want to talk about is the three domains of life. Now, there are three domains: the bacteria, the archaea, and the eukarya. Now, bacteria and archaea, those are the prokaryotic kingdoms. I mean, the prokaryotic domains. I apologize. I caught myself. I saw the word, but I was like, they're the prokaryotic domains. Now, the kingdoms are eukaryotic, and that's going to be in the domain of eukarya, you're going to have kingdom plantae, kingdom animalia, and kingdom fungi. And these are all eukaryotes. Now, plants, animals, and fungi. Now, here we have protists, which protists, they belong to many kingdoms. They don't seem to be put in. We put some of them in fungi, some of them at least right now. This may change again sometime. We had protists used to be their own kingdom. There used to be 4 kingdoms when I was a student, and I learned about Protoctista, or Protista. But now we generally count them as being part of different kingdoms. Okay, they are single-celled eukaryotes. Now, unidiversity of life. We could see that amongst life, there's a lot of similarities. If I look inside of a pond, for example, I might see a little microscopic organism called a paramecium, and they are covered in these hair-like structures called cilia that wave and beat and let it swim through the water. If I look at your windpipe, if I look in part of your nasal cavity, I will also see cells covered in cilia. It's actually called pseudostratified ciliated columnar epithelium. Now, both of these are cilia found in different things, one single-celled, one multi-cell organism, doing different jobs. But the idea that the cilia are the same and why are they there might show that there's common ancestry with single-celled organisms and multicellular organisms having those same kinds of structures that have the same common ultrastructure. They have the same common ultrastructure. They have these doublets out here, a doublet inside, a 1+9 array here. Now, they got the doublet and then the other triplets outside, doublets or triplets outside. Now, what we'll see, guys, the famous Russian geneticist Theodosius Dobzhansky said, "Nothing in biology makes sense except in the light of evolution." And so we kind of look at these weird things saying, why would this be here? Well, through evolution, it makes sense that if I share common ancestry with single-celled eukaryotes, that multicellular organisms down deep, very far back in evolutionary history share common ancestry, then it doesn't seem that surprising at all. Now, the guy who we credit mostly for this is a guy named Charles Darwin. And the fossil record was a big thing. Now, the fossil record was known by Darwin's time. People studied fossils. People understood fossils. They were trying to figure out some of these things. But this was a massive time of change, of us trying to understand things. This time frame in history was massive. You're talking about this is just before 1860. This is before this is right here coming to the American Civil War. If you think about it, when Darwin published that book, the American Civil War, which began in 1861 to 1865, those 4 years. This is just right before this. So this was turmoil in America, though. But in England, Darwin is coming up with this idea, his grand idea. And in his book on the origin of species by means of natural selection or the preservation of favored races in the struggle for life, that he wrote in 1859, is a book that articulates the ideas of what we think of as now the foundations for evolutionary theory. Now, evolution didn't stop with Darwin. It has been built on and worked on beyond Darwin. People say, well, Darwinism is dead and you need to. I was like, evolution didn't just stay still, it evolved too. Now, Darwin's basically, he goes out as a naturalist on the HMS Beagle. And one of the things on Ecuador, mainland Ecuador, he sees some birds. And those birds, actually, everybody started thinking Darwin's finches. But he sees mockingbird. And then on the Galapagos, he sees a very similar mockingbird with very minor differences. And he started to ask himself, well, obviously this thing flew from the mainland, got onto the island, and started and became an own unique species. Now, for him, he said it became a problem for him. He really wrestled with this idea and with special creation involved in that. Now, natural selection, we know now, is the mechanism that drives it. But he's like living species arose from a common ancestor. You descend from a common ancestor, then you modify from them. So we call that descent with modification. And then natural selection drives evolution. Now let's talk about why this can happen. Well, if you look out and just what next time you're out and about, though we can't see people's faces that well, we can see their eyes and we can see their hair. And when you look at the person's hair, you see that there are variations. And this hair color was inherited. Now, those variations, now also we know there's a lot of people out there. People are out there competing for mates. Now, there's usually more individuals than a population can actually be in a population that can be supported. Every generation tends to make more babies than you actually can keep. But now, in humans, we have been able to keep this high level of population. We've been able to kind of fight this. But in nature, there's a severe amount of competition. And what happens is, if I have a heritable variation that makes me good in this competition, then I'm adaptive. I always think of this cartoon about this bird, and he's like, little baby bird's asking mommy bird. He said, "Mommy, why does brother's beak look different than mine?" And mommy says, "Well, I don't want to tell you this, but your brother's adapted." They're adapted. Okay, so we get adapted. We are adaptive. We have variations that give survival advantages. Now, let's say that somebody has a hair color, an eye color, a shape, and things like that that makes them look better to females, and we're all competing out there to get a lady to love us. And then the guys who look good, they're going to get the ladies, and the guys who don't, they're not going to get the ladies, and they're adaptive, and they're going to leave behind babies. Why are they going to leave behind babies? Because if I increase the number per generation of individuals who have those, the people that are the pretty people are going to hopefully have more pretty babies and have a prettier population potentially. Now that's a very subjective, not exactly a very accurate way, but if you look at squirrels and you look at the coat color of squirrels in wild populations, it's hard to tell one squirrel apart because they all have the same basic coat coloring. Now, there are subtle differences, but it's hard to see them. Now, why that is guys, that is adaptive, and most of the population has those adaptive variations, so they are suited to their local environment. So Darwin says if a population has heritable variations, if there is competition, and if that also means if that some variations are adaptive, there will be more individuals in the population with those adaptive variations, and the whole population will be better suited. Well, this is what Darwin's things were. This is his if-then statement, okay? Now, that is evolution in a nutshell. Now, we can see this, and this is a classic example that's given. There's been experiments where they would take organisms who live in dark habitats and organisms who live in lighter habitats, and what they'll do is mix them together and put them on a dark habitat. The dark individuals tend to not get predated on as frequently because they blend in with the dark background, where the light colored individuals will get predated on. But if you switched it and you put this mixed population on a light background, it would be the darker individuals who would get weeded out. This is natural selection. There is unequal reproductive success and survival. There's greater success. Now, so it's not survival of the fittest necessarily, just those who are likely to reproduce. Now, natural selection will ultimately weed out those who are not as adaptive. So it's called natural because nature does the weeding out, and it's not because the animals want to change, they don't want to improve. So it's going to make them stand out like sore thumbs in this particular varied habitat on a black background. Now, if they got switched and you put these same varied populations on a light background, it would be opposite. And you will see that this happens. This is a great way of getting like different patches of area to have different species dominating, and even begun to create the genetic isolation needed cause speciation to happen, and changes become so pronounced that you wouldn't be able to recognize them as two species anymore. This is happening, and I've got to watch it happen in salamanders of the Appalachians, the Appalachian Mountains where we live. And I've watched this happen and see the differences, see the genetic changes happening as we speak, see the behavioral changes, see the changes in pheromones, see the changes in reproductive success, positive and negatively. And we've witnessed it, and we've been seeing it as what we predict. And that's what makes this great science, is it has predictive power. And that's with any good theory, predictive power. I'm a logical empiricist, kind of in the line of like Karl Popper. As Karl Popper used to always talk about, I do not want to use OneDrive right now. Sorry that came up. Is kind of the person is that it's a good theory if you can't disprove it is the thing. We can not disprove it. Now, so if you have a population, there's heritable variations, there's competition. The environmental factors present at that time will put a stress on them, giving differences in reproductive success, which will lead to adaptation of the population to that area. Now, those local environmental factors could change dramatically. In places like the Galapagos, there can be severe droughts because it's out near the tropics. It's a volcanic archipelago off of Ecuador. I had a very brief opportunity, and I was afraid to take it at the time, to go and be a naturalist on the Galapagos. And I almost just took off, went to the Galapagos, and just went on my grand adventure. Left everything behind. But that would have been an amazing thing to do. But I get that. Hopefully you guys are glad I didn't. I hope. Okay. Now, homology. What is this? If you look at arms, front legs of dogs, cats, whales, bats, and birds, we all have the same basic thing. We have one bones, two bones, lots of bones. You're going to see the one bone, two bones, lots of bones in the finger. Okay? One bones, two bones, lots of bones setup. This basically looks like a dog. It looks like somebody's little chihuahua with wings. I'm just saying. Now, homology is the idea that bones or structures that are arranged in a common way that were obtained from a common ancestor. Now, to be homologous, the shared common ancestry, it is structures that we have that are arranged in a common way are called homologous. But they have to be homologous. They have to have come from a common ancestor. Okay, so this does reflect their evolution. Then it reflects that inheritance is that I have a similar arrangement of forearm bones that a bat does. Okay, now homology can exist anatomically, one bones, two bones, lots of bones in whales, birds, dogs, and humans, but it also does in genes. If we think about things like trilobites, which are a very cleaner shrimp, are very simple. Insects far more modular. The modular body plans of things like simple cleaner shrimp, trilobites, even more simple. We actually can see genes being shifted to regulate certain body segments to create and adjust the modular body plans of these. And we know how that there are genetic homologies amongst all these. Like look at gene 7 being found in the same spots of all of these for most of these groups and doing some of the same things in all of them. And a shifting of the genes, it's not really making new genes to make new species, it's shifting the genes around that ultimately code for those regions and segments and adjust those regions and segments. Very fascinating stuff, guys. Okay. Evolutionary developmental biology. It would be a fantastic area to study. Also, if you're looking for a great paper topic, Evo Devo, Evolutionary Developmental Biology. Sean Carroll is a great person to look into for that. Stephen Jay Gould was kind of a big proponent of that, but Sean Carroll was kind of the king of that. And another person you might want to look at is Neil Shubin. Some of the kings of Evo Devo. Now, Darwin said there's a common ancestor, the progenitor. And this idea says if I go back, that our common ancestor for species are a long time ago. We could find the bacterial ancestors for all life. There's actually a search out there for something called the LUCA, the last universal common ancestor. But the more similarities that two species could share, the more recently they share common ancestry. Now, it's kind of like the more recent you shared a direct blood relative, the more likely, like two siblings are more genetically similar than two cousins, for example. And but that is not the way this works. But it's trying to give you like an idea of how it might work in something we're used to. Now, Darwin and his finches, this shows us an example of an evolutionary history where a common ancestor finch had arrived to the Galapagos and set up a massive adaptive radiation of these, and this is well known. Darwin's finches, people, if you want to look into, and if you get interested and would like to do this as a topic for your paper and talk about Darwin's finches as a model of evolution, Peter and Rosemary Grant are the penultimate experts on this, the Grants. One of their books, like the Beak of the Finch and things like that, How and Why Species Multiply. One of my favorite books I've read on the topic of helping me understood how the beaks changed and how these things actually went about their stuff. Cactus finches, mangrove finches, wood finches, all these finches modified to different ends to fit niches. I always said, you want to give that niche a species because niches love species. Um, so they found adaptive radiation from a single thing that gave rise to warbler finches, tree finches, and ground finches that ultimately went out and radiated more. And you're seeing that all of these can trace back eventually to the common ancestor. But there is a common ancestor of the gray warbler finch and ultimately the group that gave rise to all this unit here. this massive thing that they share a massive um, uh, uh, evolutionary relationship for that big group. Now, so now we're in science. Okay, so we can take a little break there if you like, and we're going to be in science. Now, what is science? Science is a way of knowing. Now, I want to make this clear. Science tells us about the natural world. It does not tell you guys about religious truths. It may, or at least it may help you weigh in on them, but I'm not going to say like science doesn't disprove God. Science doesn't prove God. And you can believe or not believe and accept science. Um, at least from my philosophy of science, that's how I kind of look at it. Is that science is a way of understanding. It requires logic, logos, to think, to study, and this is inferences. We infer from information. Darwin inferred right here. These are his inferences. He inferred based on what he saw. Okay, that's his inference. Inferential stuff is very important. Now, how do we get there? Well, there are many truths, but science gives us a scientific truth. Now, how do we get to the truth? Is we ask questions, we look for answers, we go around. As I always say, what does science and gangster rap have in common? It's F around and find out. And our goal here, guys, is to search for scientific truth. Now, in scientific truth, what do? Is we use a method that is our inoculation against bias, our own biases, our own stuff. The easiest person to fool is ourselves. We fool ourselves, ourselves probably are the first people we fool. Now, you have to always ask yourself, why do I believe what I believe? Why do I accept what I accept? Am I accepting the best thing? Now, in that case, guys, we use the scientific method to get through this. It helps us cut through our bias. We all have biases. Even I have biases. I try to make sure that I follow where everything takes us. So, inquiry. We want inquiry. We want to ask. So, a scientist is somebody who sees something happen and they don't just go, huh, and walk off. I can remember something I thought was really cool. I would show it to somebody. It's like, look here. And they go, huh, and they go on. I'm like, what? You don't want to know what's going on here? I'm like, a scientist sees something happen. An objective observation. We're being objective. We're looking at it very objectively, trying not to take sides, and then we start to ask questions. And I gather my information that I have. After I've asked questions, I'll gather up some information, do my research. Now, I don't mean a quick Google search. When somebody says I did my research, it's like, how long did you do it? They say, oh, about half an hour to an hour. I'm like, do you want to see all the papers that I read and exhaustively studied and analyzed and critiqued for my master's thesis? It's a lot when you're actually doing your information, and then we form hypotheses. And these hypotheses will form predictions. And from those predictions, we can test those predictions from the hypothesis. And then we analyze our data. We use that to make a conclusion. Now, here is what science does. We ask ourselves, are my results per my predictions? Are my results matching what I predict would happen? Now, you have a yes or no here. Now, that is the important thing. In non-science, in pseudoscience, in nonsense, we basically come to the conclusions and we work backwards from that, so that we put the cart before the horse and we lead it right on the primrose path. We don't do this here. We start off being objective, doubtful, and skeptical, and then a battery of tests go to it. We see, does it match what it should be? And if not, we go back and we rework. We might ask new questions. We redo our experiment. We redo our hypotheses, and we come back and do it again till we get it right. It is a self-correcting and self-adjusting methodology to prevent bias, but also repeatability. Now, at the end, if you are getting stuff, then you can communicate it. And you can communicate your results to the public and let them know. Report your findings, publish it. Until it was published, it was just a hobby. That can be a very hard thing to do. Is when everybody always acts like there's this cabal of scientists out there, I was like, who are just all working together, collaborating to come up with lies. It's like, you've never been to a scientific conference, have you? Because unlike at some kind of political rally where everyone's behind the candidate, at a scientific conference, you're not. Everybody's kind of like, I want them to be wrong. Okay? So, I always like to say scientific method is an inoculation against bias and my own what I want things to be. You can have pet peeves, but don't have pet hypotheses. You can't be married to it. You have to be ready to throw it out and start over again until you get it, okay, as you test it. Now, what I want to help you guys see, here's one that we might run into. Now, I know that at least growing up, every time the power would go out, we go get a flashlight, the flashlight be dead. Now, the flashlight doesn't work. I observed that. And you're like, why? Well, there's two reasons why. The light bulb's out or the batteries are dead. And I don't know which one. So what do? I can replace the batteries, see if that fixes it, replace the batteries. If it comes on, then the batteries were dead. So I can test that prediction. And if it doesn't work, well, it wasn't right, so I go back to hypothesis 2, and I replace the light bulb. It comes on, it's like, oh, this was the reason why, and I can test both of those. So I form a hypothesis, I make a prediction out of that hypothesis, I test the prediction, and my results should be coming along with what I predicted, okay? All right, now, in reality, we are constantly forming and testing hypotheses all the time where we're looking at how this might benefit society. We're looking at the community telling us what they would like. Community, this is what we need. Community, this is what I would like to help get involved with. We do get community involvement in science. A great example of citizen science is Partners of Amphibian and Reptile Conservation Park. They do something called the my brain just can't get it. I just cannot get that out now to save my life. I was going to, they have what's called the frog loggers, but there's something out there, and I can't remember what it's called, Partners of Amphibian and Reptile Conservation. But they have this thing, and you can do the frog loggers through that, and citizen scientists. We go out, we see, we find, and we constantly use all of this to kind of feed in our hypotheses that we form. It's more collaborative, more based on the needs as well of society as a whole. Like, we need a COVID vaccine. Okay. Now, making observations is our first step. That's when I gather my information. Once I write it down, it's called data. Information that I wrote down is called data. And that data can be quantitative. A quantity is a number. So a recorded number, like a measurement, a count. These are what you graph, chart, and tableize. You have like tables, graphs, charts, pie charts, bar graphs, things like that. Those are quantitative, counts, quantities. Qualitative is change of quality. Like if I put so many drops of biuret reagent into a solution with proteins and it turns purple, that is a qualitative data that tells me that proteins are present. Now, we still do statistics on both of these or other methods for analysis, but we always analyze these data. The data need to be analyzed. Data. I cannot make bricks without clay. It is the foundations for what we're going to do is our data. Now, our data that we've got, I have that you are seeing already preliminary stuff makes me ask questions. Like, what caused that to happen? Then I try to answer it, and I call this a hypothesis. Now, some people call a hypothesis an educated guess. I hate that. I hate that word or that explanation. Educated guess is just something I cannot stand. It is a tentative explanation to a good question. You have a well-framed question and you have a tentative answer to it. Now, what is tentative? It means I can change it. It changes with our knowledge. Now, there's actually two statistical hypotheses that we have when we do statistical analysis called the null and the alternate hypothesis. The null is sometimes H sub 0, and this is the one that says the variable I'm studying will have no effect on the data. There will be no difference between the control group and the experimental groups. Now, we haven't talked about control versus experimental group yet, but your lab hasn't now. So I need to fix that a little bit because you would have already done lab before I got to this. So I apologize. Now, but it's changed this semester. Now, the alternate hypothesis says the experimental data will affect it, positively or negative, and there will be a difference in your control experimental groups of things, and it's probably not chance. Probably is something going on here. Now, my hypotheses lead to predictions. The predictions are testable, and we can test that, and then we get our results and see if they do that. Now, experiments are one of the big ways we do this, but not the only way. We can observe. We can say, well, if this is about territorial disputes, if the male gets near the other male, they will fight. And you watch for that to happen, and you can observe it. And you made a prediction that these two males would fight if aggression was about territorial dispute, and one gets near the other one's territory and they fight. That helps to support your hypothesis. But this is why we don't prove them. We support them. You really can't prove anything in science. You can support it where anybody would be crazy not to accept it. Now, some people would disagree with me. I'm a logical empiricist. That's my philosophical underpinnings here. And so if you're kind of wanting to know a way that I think about things, kind of look at people like Karl Popper. People like that. Now, two words I want you guys to know. Deductive versus inductive reasoning. Deductive, inductive. Now, deductive reasoning, this is the most basic valid form of reasoning. Okay, this is what Sherlock Holmes did. I deduce. Now, we start very general. We start with very general statement, a hypothesis, and we go down to our logical conclusion. Like, all men are mortal. That is extremely wide-ranging, very broad. I'm a man, okay? Therefore, I am mortal. Now, all men are mortal. I'm a man. I'm mortal. All men, very general. Me, very specific. Now, the thing is, your hypothesis actually has to be correct for this to work. I can say A equals B, B equals C. So A equals C. Now, that's valid. If your hypothesis is correct, your initial premises are correct that you start off with, like all men are mortal, then you will come to the correct answers. You cannot come to the correct answers unless you start off with the correct overarching general hypothesis. Now, inductive, on the other hand, is opposite to this. It starts off specific. I have a very specific observation, and from that, I make broad generalizations. Now, from that, this is how we go from data to conclusions. This is how I form hypotheses. Now, here's the thing. We are going from specific to general, and we do this because I see a specific observation. And I can use this to figure out patterns and generalizations or explain things. I use it to craft a theory. Now, here's the thing. The conclusions can be wrong even though all the premises of the statement that I started with were correct. For example, the coin I pulled out of a bag is a penny, and that is verifiable. You could see it. And this coin is a penny. And a third coin is a penny. So I could say that all the coins in this bag are a penny. Now that's an inference, very specific. This coin, and this coin are pennies. And I saw it. Now I have a bag full of coins. It just might be there's a lot of pennies, and I drew 3 pennies. I don't know if all of them are pennies. There could be 1/4. We don't know that. Cobras are snakes. Cobras are venomous. Therefore, are snakes venomous? No, those cobras are snakes. Cobras are venomous. Not all snakes are not venomous. So I chose 2 that started off with correct statements that are specific, but created fake broad things. This is very important to understand, is that with inductive reasoning, you can get the wrong answers even though you started off with the right things. Okay? So very important. I will test on one of these 2. Absolutely. Okay? Just going to tell you that. Now, in science, we have control and experimental groups. When I do an experiment, I have a control group. Control group, basically, nothing's changed, so I can be able to compare it, and I compare it to an experimental group. The experimental group only gets one variable changed. I only alter the one variable so I can see the effect of that variable. Now, it used to be in lab 1 we did this. Now we're actually doing a yeast thing, so it's different, and I'll need to change this now, which I forgot because we changed our lab. The variables are what vary. That's what, like, there's only one variable. Like if I'm studying the effect of light on plant growth, the light level will be the variable. Now, variables can be independent or dependent. Independent variables is what I change. Like if I change the amount of light that plants are being exposed to, that's an independent variable. The treatment that the plant is getting, that's the independent variable. It always goes on the x-axis. The dependent variable, that is the one that I measure. It depends on me to measure it. It's always on the y-axis. Now, we're about done. And what I wanted to do, so let's say I have x, y, dependent variable, dependent variable, an independent variable. Sorry about that. Treatment 1. And if I graph it, in such a way, the dependent variable might be like the thing I measured. It depends on me to measure it, okay? And that is how we always do this. That is the standard for graphing. Okay. Here's an example of one where we took, this is an evolution experiment where they had stuffed mice that they put, and they had light colored mice on light backgrounds. That was our control group, the camouflaged controlled mice. And then the experimental group was the dark colored on the light background. Then we took these mice and flipped them around where we took light colored mice into like an inland habitat where it was darker colored, and the dark models in there camouflage. So the control group is always the camouflage one. And we looked at how many attacks were on there, how many actually got attacked. And we could see the control groups always were very lowly attacked compared to the experimental groups. The experimentals had more attacks. So, now, another big thing that I always like to make sure I understand, because one of the things that absolutely grinds my gears is to hear people say, well, evolution is just a theory. Well, a theory is an explanation that's broad in scope, generates new hypotheses, and has a large body of evidence. Now, theories in science are not like somebody says, I have a theory about that. They have a guess. They have more of a hypothesis than they have a theory. Now, theories in science are very different than what we use in everyday life. A lot of the times, the words used, because it was said, well, if it was a fact, it'd be called the law of evolution. No, it's not. It never will be called the law of evolution. Theories are theories because they are very broad in scope. They are products of inductive reasoning. Now, remember I said that induction can start out right and still end up wrong, unlike deduction. Now, that's the case. Theories, if it's a good theory, it can be disproven. If you want to disprove evolution, show me bunnies in the Precambrian era. Now, in the Precambrian period, if you want to show me that, then okay, then evolution's done. Evolution has not been disproven. A lot of people will say, well, we disproved it. This shows, no, it doesn't. Okay. Now, theories and laws are very different. When somebody says, what we call the law of evolution, no, they do not understand basic scientific terminologies. Laws are under very specific conditions. Like if I take a bowling ball and I climb on top of the building and I drop a bowling ball off, I could calculate how long it would take to hit the ground. Why? If I know the mass of the ball, know how high up I am, and I know what gravity is, I could calculate how long it would take to hit the ground under very specific conditions. So theories don't become laws if they grow up. Laws are made into producing theories. For example, the laws of physics were used to craft things like the quantum mechanics theory, quantum theory, things like that. But theories in biology, in some respects, one would say that a theory is more powerful than a law because theories explain the laws. Okay. It is Einstein's theories about relativity, special relativity, explain how gravity works and things like that. It is the theory of evolution is incredibly powerful and predictive. Laws can tell you how long it will take something to hit, and mathematically I can, but never fall for that pitfall. It is absolutely incorrect. It is a logical fallacy. It is people misrepresenting science. Okay. Guys, I believe this is the very last slide here, and I do like to make this point, and I don't want to step on anybody's toes too much, but science versus pseudoscience. Pseudoscience is really a big threat to people's lives. People are telling you need to take ivermectin for COVID. Ivermectin is a horse dewormer, which can harm you because if it kills worms, it also kills other healthy eukaryotic cells. It's not going to help you for COVID. The same thing goes for some of these other drugs people are trying to put. Monoclonal antibody technologies, yes, can help. The best thing you can do is go get a vaccine. Pseudoscience is peddled very heavily in anti-vaccination circles and things like that, very heavily in pseudoscience. But real science makes limited claims based on evidence. It's always repeatedly supported by evidence, and people who are using real science will put that evidence out there. But if you're making wild, exaggerated claims, I always like to say pseudoscience, that's the classic snake oil salesman. If you've never heard of a snake oil salesman, back in the Old West, some guy dressed up in a suit, his wagon would pull up, he would pull up out the street, and he's like, ladies and gentlemen, step right up, take my magic cure-all. It will cure dropsy, it cures this, it cures male impotence, it cleans your dishes, and will give your dog vigor, and all this weird stuff. And you're like, what? And it just does everything. Oh, does it help a cough? It helps a cough, sir, and everything else. It does everything. This magical, wonderful bottle here, just for a small price of 2 pieces of silver, you can have it. And what we call a panacea, a cure-all, is pseudoscience. Like aliens built the ancient world. There's absolutely no evidence for that. But all matter is made of atoms. We have never seen it not be so. Now, I will give you guys on an exam a statement that is potentially real science or pseudoscience Based on these descriptions, I will want you to be able to determine this. Okay, that is a thing I will have you do. It is like I'm letting you know. You need to be able to actually say this is an exaggerated claim. There's no evidence here. Okay, it should be generally obvious that pseudoscience is pseudoscience. Pseudo meaning false. All right, guys, that is your favorite slide. Thank you. We are done. Thank you guys for watching. This will also be on YouTube as soon as I possibly can. But thank you guys. Have a great night and a great week, and we will see you guys in the next one.